Welcome to Med Madness, where we break down complex medical concepts into easy-to-understand discussions. In today's episode, we will dive into the Ras-MAPK Signal Transduction Pathway, a critical component of cellular communication and growth. Understanding this pathway is essential, especially because mutations in Ras genes are often implicated in the development of cancers. Let's explore how this pathway works, its regulation, and its impact on health. First, let's talk about Ras proteins. These small g proteins are central to the Ras-MAPK pathway. Ras proteins exist in two states, an inactive GDP-bound state and an active GTP-bound state. When a growth factor ligand binds to a receptor tyrosine kinase on the cell membrane, it triggers autophosphorylation of the receptor. This event attracts adapter proteins that interact with Ras, promoting the removal of GDP and the binding of GTP, thereby activating Ras. Once activated, Ras initiates a phosphorylation cascade. This cascade activates mitogen-activated protein kinase, or MAPK, which then moves into the nucleus to influence gene transcription, impacting cell growth and division. Ras proteins have intrinsic GTPase activity, which allows them to hydrolyze GTP back to GDP, turning off the signal when it's no longer needed. This regulation is crucial to prevent excessive cell proliferation. However, when there are mutations in RAS genes, this intrinsic GTPase activity can be impaired. As a result, Ras remains in its active state, causing continuous and uncontrolled cell proliferation. Such mutations are commonly found in various cancers, including colorectal and pancreatic malignancies. For context, other molecules like ATP, CA2+, and IP3 play essential roles in different signaling pathways. ATP provides phosphates for kinase reactions, Ca2+ is vital for many cellular functions, and IP3 works with DAG in another signaling system to regulate intracellular calcium levels. Let me illustrate this with a brief case study. Consider a 45-year-old patient diagnosed with colorectal cancer. Genetic analysis reveals a mutation in the KRAS gene, one of the most commonly mutated Ras genes in cancer. This mutation leads to a constitutively active Ras protein, driving the uncontrolled cell proliferation seen in the tumor. Understanding this mutation helps in selecting targeted therapies that can inhibit the downstream effects of this aberrant signaling pathway. To wrap up, we've covered the basics of the Ras-MAPK Signal Transduction Pathway, its regulation, and its role in cancer when mutations occur. Remember, this pathway's regulation relies on a balance between active and inactive Ras proteins. Mutations disrupting this balance can lead to malignancies. Engage with our pre- and post-podcast quizzes and use Anki flashcards to reinforce today's topic. These tools are designed to enhance your understanding and retention of complex medical concepts. Thank you for tuning in to Med Madness. Stay curious and keep learning.